All right, well, thank you very, very much for the kind introduction, and I also wanted to thank the organizers, the session chairs, and the speakers, and the participants for what has been, for me at least, a really, really interesting meeting and a very inspiring meeting. So my talk today is titled Stories from the Supplement, and let me begin by telling you why. So as many of you know, I work on RNA-seq recently in the last few years, and we wrote a program called Cufflinks. And Cufflinks was published in 2010. It was a collaboration between my group, the group of Steven Salzberg and Barbara Wald. And when we wrote the paper, it was a methods paper for a method for assembling and quantifying RNA-seq. We wanted to describe the method, and we really couldn't do it in the journal. Nature Biotechnology, like many journals, all journals really, in this area don't publish really the methods in the paper. So we wrote a 42-page supplement. And it's actually a LaTeX file, for those of you who know what that means, and it's full of math and our method. It upset me at the time because it was never reviewed. The only person who really, really carefully checked the supplement was actually Steven Salzberg. He saved table 1. He gave us feedback and helped write some of it. The reviewers didn't really ever give us feedback on the supplement, and it has errors. I was going to offer today to pay people who find the errors, but I won't do that. There are too many. And you know, from my point of view, review is not an obstacle to publication. It's a good thing, and I wanted the thing to be reviewed, and it wasn't. The other thing is, you know, what's in the supplement is not just some description of a method. It's actually ideas that enable biology. So one of the things I'm proud about the Cufflinks paper is not the software per se, but we introduced some ideas. The idea that you can look for changes in isoforms by doing RNA-seq. We introduced the square root of the Jensen-Shannon divergence, which is an idea from information theory, a metric on probability distributions that we didn't create, but that wasn't in this field and is very useful in this setting. And so all these ideas are buried there, and I doubt very many people actually read it. In 2012, we published what's the Cufflinks 2 paper, where we really tried to address deficiencies in the method in differential expression. We didn't really have time to do that for this paper, but we knew we needed to work on it, and we were working on it. And then we published it, and at this point, the supplementary material was 70 pages. So we have a lot more math and lots of figures, and this paper described the collaboration with the RIN lab, described a large experiment as well. So at that point, I was writing my third RNA-seq paper, method paper, on a program called Express. And I was wondering what I should do. I looked at these numbers, and I thought in the arithmetic progression, the next supplement ought to be 98 pages. But it gets a bit ridiculous, so I thought to myself, OK, I'm not doing that. And what I did instead is I worked very hard with my student, Adam Roberts, to write the methods in the methods, the online methods. And we got it down to about 7 or 8 pages of online methods. And then we got the paper reviewed, and the editors, both of these papers actually, in this case, Craig Mack and Tal Navi, were very helpful, got us very nice reviews, really worked with us. But then the sort of editorial staff at the final process sent me this. And this is actually their highlight. I didn't highlight this. They said you're allowed only 10 equations. So there was no arguing this. It reminded me of this famous quote from the movie Amadeus. And I'm not comparing myself to Mozart, but I am comparing nature methods to Emperor Joseph II, who said to Mozart, you have too many notes. You know? So we had to cut. So I went through the mental exercise of reducing the methods to 10 equations. That's a pretty hard thing to do for RNA-seq. This is the final online methods, and there's equation number 10. I will show you during this talk that this equation, which is the key equation for the paper, I mean, it's not understandable from this online methods. That's not my fault. That's all I have to say. And I just wanted to give this talk today to tell you some stories from the supplements in my papers, and partly to make the point that I think this is a really terrible, terrible thing for our field. It's limiting the ability for computational biologists to have impact to write good papers, and it's just a terrible thing. So I would like today to talk about what's in the supplement. I'm going to tell 3 stories very quickly. The first is an insight from our supplements that took us a few years to really get to the bottom of, but that's that fragment assignment. The issue of dealing with ambiguously mapped reads is really crucial, not just for RNA-seq, but for all high-throughput sequencing experiments. And the idea here about dealing with ambiguously mapped fragments actually goes back to the original RNA-seq paper by Ali Mortazavi, where he invented a method called rescue. And this is part of figure 1 in his paper, where he talks about dealing with ambiguously mapped reads. But I think at the time this paper was published, at least for me, I didn't really realize at the time the really key importance of these reads. It seemed at first that maybe you could just throw them out or ignore them. But because RNA-seq involves multiple, quantifying isoforms from genes, in many cases even dozens of isoforms that overlap, really many, many reads are ambiguously mapped to the transcripts. They may map uniquely to the genome, but in terms of the transcripts, they're ambiguous. So that's one thing I want to talk about first. I'd like to say for a moment what I mean by star-seq and not just RNA-seq. These are a whole collection of assays where you would like to measure something, you reduce it to sequencing, and then you go and sequence. Now, I'm talking about ChIP-seq and methyl-seq and lots of the assays we've heard here. And one of the things I've been working on in the group is to try to sort of figure out what commonalities these assays have. And I can make this cartoon now, I think is pretty fair. You'd like to measure something, you figure out how to do that by sequencing. Then you have to take the reads and figure out what the measurement was. And this is a key problem here. This is part of the technology. It's not just something you add on at the end. And often this part is very non-intuitive. And it's math and statistics. Then you do the actual analysis, and that's the interesting part often. But how you do this part affects what you can do here. So the first assay was ChIP-seq. I think that was published of this kind. It was converting ChIP-ChIP to ChIP-seq. And here are the steps. This is for looking for protein DNA binding. At the end of the day, you end up with looking for actual motifs. RNA-seq is very similar, except as we all know by now, you're doing something very different. You're looking for RNA abundance in this case, and you're sequencing fragments from the RNAs. At the end of the day, you're interested in a pie chart, which is how much of each of the transcripts was in the pool that you sequenced. And the actual application of this downstream involves many things. I call them differential analysis, not expression, to capture the wide variety of tasks, such as figure out what's happened during development, or find EQTLs, or whatever it is. This involves comparing different samples. But to do this, you need the pie charts. And so that's the problem in RNA-seq. There was a paper published recently highlighting many, many methods. And they're not, they're just really a wild zoo of ideas. There are methods for doing geometry of, chromatin, looking for structure of RNA, binding of various kinds, translation, transcription, and the imagination is the limit. In fact, I've counted more than 50 such assays. I know of actually 52 right now, and I'm cataloging them. The wildest by far is actually from outer space, is a method to detect weakly interacting massive particles. As a proposal, it's not an actual, it's not been implemented. It's in an archive paper. It's got George Church as a co-author, and I sort of take that seriously, okay? So, and their idea is something like hanging single-strand DNA from some matrix and have the particles, smashing them, cutting them, and then you sequence them, and you see how the particles pass through Earth. So I think this is just to show you that really there is probably a lot of application of sequencing we haven't even thought about yet. As you've heard, I've started an infamous blog, and I'm maintaining a page there that lists all the assays. And one thing I'd like to ask for is if you see any that are missing, please let me know. I'm currently at 51, and I'll be adding ICE-seq tonight or tomorrow. And there's also ATAC-seq is a really interesting new assay that I think is very good for basically replacing DNA-seq. So what is the inverse problem that we're talking about? And that's the focus of my talk, is the method. So it's all good to sequence and get data, but you need to go back and figure out the measurement in all of these assays. And one insight that we had, and this is with my student Cole Trapnell, who's now a postdoc at Harvard, my former student Adam Roberts, who's just graduated, is after, this seems kind of obvious to us now, but we eventually realized that one way to think about this whole problem is to realize that there are two fundamental issues that occur in every assay. And they are what I call fragment assignment and density deconvolution. Fragment assignment is the problem of figuring out where the reads really came from. And density deconvolution is for figuring out in every site of your target might be a transcript, but it might be a genome in ChIP-seq or something else, figuring out how many reads were actually in that position on the target. So density deconvolution is like figuring out where the peaks are in your ChIP-seq. And fragment assignment is figuring out where the reads go. So this is sort of a chicken and egg problem, because if you know where the fragments truly came from, then it's trivial to draw the coverage plots of how many reads were at every site. But if you don't know where the reads came from, that's hard to do. Okay? Conversely, if you really know how many reads came from this site, it's much easier to figure out where the read came from when you don't really know. So these problems go hand in hand. And I think for many assays, ChIP-seq being a great example, we heard a little bit about, there was a very interesting talk at the meeting I think yesterday. People have kind of ignored ChIP-seq peaks in repetitive regions, and we've also been working on this, and there's a whole world of binding to find there because you need to just simply solve these problems together just like in RNA-seq, and in fact the same tools can solve them. And so that's kind of what we've been working on. So let me just talk very briefly about the fragment assignment problem. And what it means. So in the simplest case, you have 3 fragments, and you're going to pick a read out of the fragments, sort of generatively speaking. And what you measure are, I call them Qs, are the number of green, red, and blue you saw. And in my first thought experiment, there is no ambiguous mapping. Things are nice and simple, and you really know how many reads came from red, green, or blue. And the goal now is to figure out what the pie chart was, and the naive thing to do and the correct thing to do is to take the ratio of blue over red, green, and blue. And one thing I think people often forget is that all of these star-seq assays are inherently random. So the read numbers here are random. If you replicate the experiment, you're going to get different numbers here. So this can only be an estimate. The pie chart is fuzzy, okay? And that's important to remember. But the real problem we're dealing with involves having ambiguously mapped reads. Cyan is a mixture of green and blue, and there's purple, yellow, and there are actually going to be black reads that just they could have come from anywhere. And so really what the data actually looks like is that it involves counting not just the number of uniquely mapped red, green, and blue, but all these other combinations. Now you can just ignore this and throw it out and get an estimate for this. And actually, that's not a terrible thing to do, except that by throwing away a lot of data, you greatly increase the variance on your estimate because, as I said before, these numbers are random, okay? They don't look random to you because you have a set of numbers, but they're random because if you repeat the experiment, you get other numbers. And so your estimate will have a very high variance. It will be noisy. So how does one actually do this? Well, I just need to tell you one more thing that's really, really fundamental in RNA-seq, and people have made really embarrassing errors about this issue. There are actually 2 pie charts. There's the pie chart that I call the row pie chart, which is really what I meant to talk about before, which is how much of blue, green, and red is in your sample. But there's the alpha pie chart, which is what's the probability of picking out a read from red, green, and blue. Now those sound kind of the same. If these are the abundances, surely picking a readout is the same probability, but it doesn't, because if red is long and green is short, then you'll just be more likely to pick out of red because you're picking randomly, okay? So the longer a transcript is, there's just more chance you pick from it. So alpha is basically like the row where you normalize for the length. And it turns out, it's not hard to see that the 2 pie charts are kind of equivalent in the sense that if you know this pie chart, you can recover this one by performing some inverse operation with the lengths, and you can go from this one to this one. They're equivalent, okay? So it doesn't matter which you work with, but it turns out that in RNA-seq software and methodology, it's very convenient to talk about these and not those. So many or most of the programs work internally with these and not with those. And that's what I'm going to do. So what you do, the basic principle behind all of RNA-seq analysis is that you write out the probability for seeing your data, and it's basically the probability that you pull a read out of the transcript times the number of times you saw that thing to the power of that. And so the idea is very simple conceptually. You try to find the pie chart that makes it the most likely to have seen your data. One thing that happens in RNA-seq that frustrates me is people argue with me a lot about, oh, I just want to use counts or this or that, and I'll talk more about that in a second. But it doesn't matter what they're saying. They're doing something like this, but just they're picking a different model, okay? So that's basically what's going on. And the goal, there's some math here, but it says basically that this is a very nice function and that you can actually maximize it. And as I speak, my 250 freshmen back at Berkeley at this moment are taking their second midterm and doing this kind of thing, poor freshmen. So the algorithm, the rescue algorithm that Ali wrote about in his paper is what's known in statistics as the EM algorithm. And as far as I can tell, it goes back to a paper by Chris Lee from UCLA looking at ESTs actually in 2006. He thought about this EM algorithm in this context. The idea is very intuitive. You start with a pie chart that's uniform, where you say, I don't know anything, and then you split your reads according to that pie chart. So, if a read mapped to red and blue, then you send half of it you give half a count to red and half a count to blue. It's kind of an intuitive thing to do. And once you've done that, you sum up the counts, and you figure out the fractional count of red, the fractional contribution of green and blue, and you build a new pie chart. And now you split your reads according to this new pie chart. So you're kind of solving the chicken and egg problem by doing chicken and then egg and then chicken and then egg. And that's called the EM algorithm. And it is a nice mathematical miracle that it converges, actually, and actually maximizes this function. And there are other ways to maximize the function, but this one is simple and intuitive. And many RNA-seq methods implement this. And at the end, you get a pie chart. Now, one thing that people often tell me, since a lot of people talk to me about RNA-seq, is they don't feel comfortable with this because it's a method. They would prefer to just look at the reads. You know, the reads are real. They have sequences in them, and they're really good and real. But this feels like who knows exactly what this is, about hokey pokey. But the thing is, right, that it doesn't matter. Even if they don't do this, their reads were random. So I don't really understand that, but that's how it is. OK. This is the actual method in Express, is our software. And it's very similar to cufflinks, although I think it's better, this particular likelihood function. And it's also very similar to RSEM and a number of other programs. And the difference is that in the model I just described, things are very simple. The model is that transcripts have uniform coverage across the transcript, and that you really just select out at random the reads. But reads really have error, and there are biases. And these models try to capture that, that's all. And so there's more variables. And I'm not going to explain this all to you. I don't have time. And unfortunately, you cannot read about it in the paper either, because it was eliminated. That equation's right there. It's number 3. So I only got 2 equations before it. So I'm sorry. Where it is now available is it's published just now, like literally a few weeks ago, in Adam's thesis. So Adam's PhD thesis, which I will put online shortly, has all the details of this method. But it's unfortunately not in the paper. So much for peer review. What I want to say is that why do I think it's important to think about these things? Well, if you think about something deeply, then you actually have insights that may not have been so obvious otherwise. And my student Nicholas Bray and I have been thinking about this and had an insight. So that comes from thinking about fragment assignment, and that I think is actually useful. So I'll tell you about that. So no sample is an island. What's happened in RNA-seq is that experiments no longer involve the naive condition A and B, and I did 3 replicates in each. What's actually happening, there are large consortiums. Just published was the GVADIS, I think it's pronounced, consortium, publishing 462 individuals from 1,000 genomes RNA-seq on LCLs. There's a big GTEx consortium looking at tissues in many, many different tissues in cadavers. They already have 2,000 or so RNA-seq samples, with 20,000 coming very soon in the next few years. There are developmental transcriptomes. This was published in 2012, landmark paper, but many more coming. And single-cell RNA-seq, by the very virtue of the assay, involves doing at first a couple, this year hundreds, and next year thousands. I know already of a number of experiments with 10,000 cells that are going to be published shortly. There was a paper last week with about 100 cells. So all the experiments have tons and tons of essentially different RNA-seq from related things, in this case, related people, related cells, related stages of development, but different. And it's kind of weird. The idea of replicate, which a lot of the statisticians care a lot about, it sort of breaks down in this context in the sense that every single experiment here is, in fact, a replicate. If you're doing 2 stages, even if they're just a little bit apart, one is sort of a replicate of the other, just tweaked a bit. So you kind of have this continuum between really true replicate and actually different condition. It's not either or. So we've been thinking about what to do about this. And our insight is that these experiments, they're kind of like the islands of Hawaii. On the surface, they look like they're just different experiments. But underneath, they're very connected to each other, because the actual transcript abundances in these different samples are mostly the same. A few things change. You're interested in what changed, not what's the same. But in large part, there's a commonality to the structure. So we looked at the EM algorithm that I just explained to you. Currently, you take every one of these pie charts, and you simply run the EM algorithm, or you just count, or whatever. But you just do every sample independently. And all the papers on single cell so far did that. And that is, in fact, how all the consortiums are doing their analysis. They're just taking every sample, doing something to it, and then they go and do analysis. But instead, you might think to actually model the joint structure. So in this case, I have new pie charts. They're yellow and purple. They're kind of master pie charts. We're kind of imagining that these little pie charts come from 2 real different things. And there's somehow mixtures of these things according to these different mixtures. That's a model. Instead of 2, you might have 3, 4, 5, and we can talk about how to choose that. But the general idea is to select out representatives, kind of archetypes of your experiment, and then consider your samples to be mixed. So in the case of development, you might imagine that you have kind of landmarks along the developmental time course. Those are your archetypes. And then in between the various experiments you're doing are interpolations between those. Well, what you can do then is instead of doing this back and forth rescue here, EM, you first go and figure out the representatives. And then using these, you assign your reads. And you kind of iterate a more complicated process of estimating all 3 of these things at the same time. There's an interpretation to this and a methodology that's sort of more rigorous than the intuitive explanation I've given. You can really think of this as, if this is your matrix of transcripts experiments, you're modeling it like as decomposable like a product of 2 low-rank matrices. And in machine learning, that is how the Netflix prize was won, in fact. It's a very powerful technique to take large matrices and reduce them to a low-rank approximation. And this is done by something called the Lee-Sung algorithm. That's one method. And it is exactly what we're actually doing here. We're finding the best approximation of this type to our data. It's really exactly the Netflix situation where you have different movies and users, and you're trying to figure out what people like by sort of a model of sharing between the users and the movies. Okay, that's exactly what we're doing here. What's really nice is that this algorithm, it gels very nicely with this one. And I don't have time to explain the details, but there's a master EM algorithm that really solves this problem optimally and ends up being, in an implementation sense, really trivial modification of the standard algorithm. So we did it. This is not published yet. It's work in progress, but I'll show you a quick result. We ran the GVADIS data set. So we ran Express on the data set because it's our program and fast and we think fairly accurate. This was just published, I think, 2 weeks ago. And it has 30 million reads per sample. So what we did with it is to take just 1 million reads, or even 500,000, out of this 30 million, run our method, and ask, how does the accuracy we get with just a small sample from every individual compare to what we get when we actually take the 30 million reads and quantify the transcriptomes? And I think the results are really encouraging. So this is the correlation, the rank correlation. Maybe it's not the best, but a standard measure in RNA-seq. And these are the number of reads. And so what you can see is when you do things independently, which is what people are doing, your accuracy improves as you have more and more reads. The reason people pick 30 million is they think that that gets you most of the stuff, maybe. I mean, that's a debatable number. But that's what they did. There's a budget limit. But what you can see is that the estimated values are given by blue and red. Now, I'll just explain briefly. Red will make people very uncomfortable. That's where we actually impute the actual expression level. Now, if statistics makes you queasy, well, you can instead just take our method and then actually just give sort of as a prior what we've actually think the abundances are of things, and then just run 1 round of rescue, and that's blue. So we believe in red, but that's blue. And what you can see is there's really an order of magnitude difference. So for example, with just 500,000 reads using this method, you're essentially getting an accuracy compared to 4 million reads, or 4 or 5 million reads. So you might ask, well, what happens if you put in 30 million reads? And I think the answer will be that you'll be doing like you had 60 or 70 or 100 million reads. And I don't know the answer yet because we haven't done that experiment. We really need to go somewhere where we have very, very deep sequence to really check this carefully. But there's no mathematical reason why this doesn't magnify any experiment. And so we will hopefully have a paper on the archive about this very soon. The second story I want to tell is about counting reads from the supplement. I have to tell this story because a lot of people are doing RNA-seq, and many of them, I honestly believe, are just doing a wrong thing. And I go around giving talks, and I make this point, and less people do the wrong thing. So I'm going to hopefully convert 1 person here. I'm happy. And it's a nice story from the supplement. So this figure comes from our Cufflinks 2 paper, and it involves the idea, an analysis of the idea that statistics is not good for RNA-seq, and you should just count the reads. What that means is, well, how do you count the reads? Well, there are different isoforms, and you don't really know reads that are ambiguous to this exon. I mean, you don't really know which transcript they came from. So people propose different ideas. Well, they say, maybe you take exon union. Just take the whole genomic region, add up the number of reads. Okay? And that's your proxy for the expression of that gene. Another idea is to say, you know what, maybe that's not so good. Let's just look at the stuff where they share sequence in common. Here you would do 4 reads instead of the 12. I mean, the argument here is that, well, maybe I have less data, so I have a bit more noise, but this is a good proxy. I'm really just looking at what's common. So obviously, that's the total sharing that's happened. I'm going to argue to you that this just doesn't work. And the math is what I'm also testing my students on. Well, here's the math. When people are suggesting that, they have an intuition in mind. And it just happens to be it's wrong. So here's the intuition. They say, OK, adding up just the counts, like taking the total 12 here, yeah, maybe it's wrong because these have a different length and you maybe need to normalize by length. We agree it's wrong. But we're not actually caring about expression. We're biologists, and we always have controls, and we're doing 2 different conditions. Like say we're doing differential expression, or if we're doing an EQTL, we're doing a regression. So really what we care about is just the ratio. And if the error, yeah, sure, we're introducing some bias, but it's the same bias, right? And so it just cancels out. And that's like saying, and this is not just a metaphorical analogy. In fact, I'll show you in a second. It's actually, in fact, exact analogy. Is what you're saying is that you're adding a half plus a 2/3 by what I call, being at Berkeley, I call this the Stanford method. You add the numerators and the denominators. So you get 3/5, and then you take the bottom here, and you get 8/10. And then you say, yes, 3/5 over 8/10, that's 3/4. And I've canceled the error out. It was wrong on top and wrong in the bottom, but it's canceling out. And that's just not true. Okay? Sometimes things are true, sometimes they're false. And in math, it's one or the other, unless you're a logician. So the answer here, I'm sure you all did it in your head very quickly. It's actually 0.7368. It's pretty close, but it's not 3/4. And you're either right or you're wrong. One of the arguments I get from people is, OK, but 3/4 is pretty close to 0.73, so who cares? Okay. So that's what we actually went to look at in our paper. So first we have this figure that actually just does this in a thought experiment, but with actual reads on transcripts instead of the fractions I showed you. And basically computes the things here. And it turns out that we can, in a thought experiment, make anything happen. So even the intersection method, where you really intuitively, it just feels right, like things cancel, they don't. And we can have it so that the log-fold change is actually positive in one thing, but in reality is negative. Anything can happen. You can get the totally wrong qualitative result. Okay? So I won't go through this right now, but if you want to really think about this thought experiment, this is in our paper and also on my blog. What we went out to do, thanks to John Rinn, who is Cole's postdoc mentor, they went out and they did a whole bunch of experiments. And I think we actually have a very nice data set. If people want to actually benchmark and look at RNA-seq, sometimes people tell me there's no data. Well, there is. We did a large RNA-seq experiment. It's in a very interesting biology study I won't talk about. But there are 3 replicates of the RNA-seq for every one of the conditions we did, and 3 replicates from the same RNA of an array experiment. So we have microarray data and RNA-seq data. And what I'm showing here is that, in fact, when you take the intersection count, you just look at the intersecting parts, and you quantify expression that way, and then you compare it to the actual array fold change, then things look better when you do the right math. So in every point here, the top, I should say this end of the arrow is the naive method. The bottom of the end of the arrow is the expression estimate from cufflinks or cuffdiff. And what you can see is that, the point of these arrows is that blue is going up and red is going down. Okay? So you can just see, looking at this picture, that you're improving the naive method when you actually do the right thing, as quantified by arrays. Now, we actually hope that RNA-seq is more accurate than arrays, but I hope you believe me that this is a good thing. So on real data, doing the right thing actually improves results. Now, one of the things is, you might say, OK, but does it really matter for differential expression? Yes, it does. So we actually checked that with DE-seq, which is a popular method, the fold change is simply inaccurate. So this is the same picture as before, but not the naive intersection, but actually DE-seq's method. And it's the same story. I'm sorry it's not colored in red and blue, but all the arrows that are on top here point down, and the ones on the bottom are pointing up, and the rank correlation, I don't have it on the slide here, but it improves greatly. And one of the points I like to make is that in analysis of RNA-seq, it's not always the p-value at the end that's important. The fold change is often the actual quantity of interest. So even if DE-seq, at the end of the day, says the same thing as differentially expressed, which it doesn't change the fact that the fold change estimate is just wrong. And so you don't understand the actual true fold change. So that was my second story. I want to end soon because I'm standing between you and dinner, and so I'm ready for my third story. And for that, I want to set the record straight on something that is a current complete mess in RNA-seq. And for those of you who are tweeting, please tweet this. OK. So units for RNA-seq. At this meeting, I heard CPM, counts per million. That's where you just add up all the counts. Somebody was doing counts per million. I've heard RPKM. That was introduced by Ali Mortazavi. I've heard FPKM. That was introduced in our paper. There's another unit called TPM, transcripts per million. I think I heard that one as well, in fact, at this meeting. That was introduced in the paper from Colin Dewey's group at Wisconsin, publishing RSIM. And the question is, are they all the same? Or is one better than the other? Or what should you actually use? And I think it's fair to say that people don't really know what they're doing on average. This is a quote I pulled out from Seq Answers, and I've highlighted in red everything here that's wrong. So this is a person saying something and a person answering them, and they really have no idea what RPKM is. And in fact, I see this all the time. So not to be negative, but really, people kind of just use one of these things because the programs output it. But they're not the same. Let me explain briefly what they actually are. First of all, RPKM is not a method for quantifying expression. It's a unit. Okay, that's an important thing to understand. The abundances, I just want to remind you, I'm representing by row, and please remember these alphas are kind of the probabilities of pulling reads out of the transcripts. So here's an analysis of what happens in converting from the row to the alpha. You see, row, it looks like, remember, it's the alphas normalized by length. But in the really naive model, I'm only going to do the simplest case here today. I already showed you, like, if you want to measure the abundance, you just take the number of reads, and you divide it by the total number of reads. Okay? That was remember the simple red, green, blue. Just take the number that are red and divide by the total. So the hat here, that's your estimate. That is the kind of best estimate. There's a reason why it's good, but it's also intuitive. Just take the number of red over the total. But if you parse through the math here, what you see is that there's still a remaining length term. In the original RPKM, this length didn't have that little squiggle. The squiggle comes from the fact that there's an edge effect. When you do RNA-seq, you cannot actually pull a fragment right out from the very end because there's a size selection, and some fragments have a minimum length. So that's a detail. It's not so important. Then there's the denominator here. But you see the denominator, it's actually fixed. It doesn't depend on t, the actual transcript whose abundance you're calculating. It's like a normalization constant. And so you can just ignore it and put a proportionality sign. Okay? So you're just taking the total number of reads at the denominator, the length, the number you saw that are red over the total, and you're putting that here, and you throw this out. That's why it disappeared there. Now, there's also a 10 cubed and a 10 to the 6th. And I'll introduce that because if you didn't put those numbers there, then things look tiny and weird. And you don't want to go around reporting 0.00003. So you add in the 1,000 and the million, so you have nice big round numbers. And that is RPKM. It's said to take the number of reads from the transcript, divide by the length over 1,000, and the total number of reads mapped over 1,000,000. And this can be extended to doing RPKM even when reads map ambiguously. You just have to replace the alpha hat by a more complicated thing that you estimated, but this remains the same. So that is actually what RPKM is. It is actually like the row. You see, it's the same thing. It's proportional. So it's like the row, but you just moved it all by a scalar. Okay? We introduced FPKM because we felt Ali hadn't thought too much about paired end reads. It wasn't his fault. There weren't many around at the time, or any. But really, the actual thing you're sequencing is a fragment. You're sequencing the ends. And so it's more accurate to speak of FPKM, but that's the only difference between RPKM and FPKM. And I like this, actually, and I think it was a good unit, but I think it's the wrong unit. I hadn't thought it through carefully when we published cufflinks, but nobody read the supplement, nobody reviewed it, and so nobody pointed it out. And when we published it, everybody started using cufflinks. So now, every time I open a paper and it says FPKM, I feel awful inside. And I scream and yell and shout at my students. OK, so why don't I like FPKM? Well, here's the thing. This proportionality constant depends on the actual experiment. Although, in a fixed experiment, the rows here, the true abundances, are proportional to this thing. If you do a different experiment, this factor changes because it's got the Q in the denominator there. So the normalization factor actually depends on the actual abundances of the experiment. So if you do 2 experiments, and you're doing FPKM for both, then for each of them, the FPKM is proportional, if you took the sum of the FPKMs, to the abundance. But the proportionality constant is different. It's not apples to apples. So it's not a good idea to do this. Okay? Now, we just didn't think about this carefully, because when we did the cufflinks paper, we didn't have another replicate or another condition. We did myogenesis, and we were just assembling a data set and reporting the results. Like there was a time series, but we didn't really think very carefully about this. And so it's not actually a good idea to use this unit. A better unit is just, and this is kind of a bit embarrassing, to use rho. That's what you care about. Why don't you just use it? And that is actually exactly what TPM is. And that's what sort of Colin Dewey tried to say in his paper. So he just said, just take rho and multiply it by 1,000,000 because otherwise the numbers are 10 to the minus 5th. So just take the rho. But 1,000,000 is constant across experiments. So that doesn't mean, I want to say, that you can just compare exactly the TPM values across experiments, because there are other things that change. And you do need to normalize. And it's good, for example, to change this, normalize this n. People basically take the median or something like that because they want to be robust, so like a very highly expressed transcript and so on. But basically, this is what you want to do. You just want to take the rho. So one thing I think people ought to do is use TPM in their papers. So those are the 3 stories I wanted to tell. I have one very last thing to say, and then I'll stop, and we can go to dinner soon. I hope you've seen that there is useful stuff in the supplement. Not just methodology that tells you what the method is doing, but ideas. Ideas that actually enable new biology. Ideas that could lead you wrong, could lead you astray, and ideas that could lead you into good places. And one of the things that frustrates me is this plot. I believe it was shown here on the first night, but I got here late. I've seen many of these plots. I also see plots about the number of reads as a function of time in every talk nowadays in genomics, almost. And what I don't like about this slide, well, it's actually a very interesting slide. There are sequencers where I grew up in South Africa, and you can see that people in Boston have bought a lot of machines, but China is winning. Why don't I like this slide? Well, it's about the data, okay? All of these slides, they just show you the data. And the data is, from my point of view, very important, very interesting. But where's the method? Nobody's talking about how many bioinformatics researchers there are in these places. Nobody's talking about how many papers in computational biology and RNA-seq are being written per year, okay? So I really don't like this slide, so I fixed it. And I made the cufflinks version. So we have a little Google Analytics on the cufflinks website, and this is where people have been using cufflinks, okay? I actually think there's sort of a fundamental difference in these plots. They look very, very similar. This plot, though, is showing where people are actually coming to our website and reading it and downloading the software and doing analysis, okay? My point here is that it's actually just the same. And it's just as incredible and amazing, the increase in the number of people doing this. Everything that you've heard about data is also true about the methods and the analysis. And that's why it should be up front and center. What I think is, I'm sorry? It's a good point, but I think I have a comment about that question that was shouted out, is that I had a slide I started to prepare, but I decided not to show it. I think the right thing to normalize by is money. And I actually calculated how much it costs me per graduate student doing the methods versus per run through the HiSeq. And it's much more expensive to do the analysis. And therefore, it's much more relevant, actually. So that's the right normalization factor. Anyway, I like this plot. I've superimposed the 2 on each other. So this is the great opacity feature. And you can actually see great agreement, but not always. In Colombia, they apparently, I don't know, maybe they do have a sequencer, and that plot was outdated, but they're doing a bunch of analysis in the Columbia Genome Center. I looked it up. In Russia, although there's a sequencer there, the analysis is being done in Novosibirsk. And you can basically see that it's sort of a very interesting plot, and it's interesting to dig down into this. But I will stop with that and just finish by saying that I think methods matter. Thank you very much.
Thank you very much for a great talk. So there is one solution to your maximum of 10 equations per paper, is that you have to publish in a different journal.
That's a good point, and I have done that. And I have published, I'm in the mathematics department and the biology department, and I've published in math journals and in the archive. But that's not so simple as it sounds. And the reason is that, first of all, different publishing venues have very different criteria. So let me give a simple example. In my mathematics department, there is a sign on the board outside the secretary's office of the chair that says all papers in this department need to have authors in alphabetical order. So when I publish with biologists, I simply can't ask them to put everything in alphabetical order. Now that's a problem because when you publish in a math journal, if you have 20 authors on your paper, that doesn't work out. The editors are confused. It also takes about 2 years from submission to publication. That's partly because people actually read the papers. It's not a bad thing. But that's one issue. And I think it's a very good point. Another issue is that I actually believe that journals like Nature and Science play a very important role. They do something that math journals, for example, don't do a very good job of. I've found that the editors I've worked with are very, very helpful in actually editing the paper with you. And that's something that doesn't happen in many other venues. So yes, I do publish elsewhere, and I try to put a lot of stuff in the archive. But I don't see why there should be any journal that thinks it's okay not to publish methods. That just doesn't make any sense. Yes?
I like the idea of holding samples, but do you think it's going to, how is it going to scale with increasing number of data sets?
That's a very good question, and I haven't gone through this, but the algorithm is fairly simple. From the point of view of, it's basically equivalent to the standard RNA-seq kind of process with the modification. There's a very good point you make that you will need very fast algorithms for RNA-seq, and that's why I worked on Express. So in around 2011, I started getting email. It was actually in the context of the Drosophila paper doing the developmental time course, and they were having trouble running cufflinks a little bit, and so they were asking us for help, and we did help them. But I thought to do Express, because I wanted to be fast, and you heard a great talk at this meeting about a program called Sailfish that's even faster. And that's one point I want to make, that people often think, okay, it's faster, who cares? But the point is that a faster program enables stuff like this. And so it's actually very, very important to be fast because it enables entirely new ideas.
Thanks, great talk. I was just curious if you had any suggestions about how you think as a community we can really sort of bring about the kind of changes we need to make people realize that methods matter and that this kind of stuff shouldn't be buried, and that we should really push this to be upfront and as important as the results that are reported, because that's how the results are created.
I've thought about this a lot, and I don't have a good answer. The truth is, it's why I started my blog. I felt that I didn't have a voice other than to just try to go out on the internet and make a case. I've talked to editors. I've talked to editors both at Genome Biology and Nature Biotechnology, and other journals have told me that they themselves feel there's a problem, but it's difficult to change it. I think that one thing that has to happen is there needs to be first a coalescence around what the standards even ought to be. And I'm working on a paper now I'm writing where I make the argument that just a very simple example I'll give is that, there's this idea of benchmarking a method. And it's very vague right now in the literature, and there's actually a huge difference between, so in statistics, what people do is they run simulations. But not because that proves the method is good, but it actually proves that the software correctly implements what they wanted it to do. In other words, they use simulation to prove they're not wrong, not that they're right. In biology, there's an emphasis, obviously, on real data. That's equally as important. But if you require both, your actual bar for a computational biology paper is much higher than any other field I know of. So that is actually the case, and I think that should be done. But then if you're going to attain a higher bar, then the journals have to agree to publish the methods as a paper. So I don't have a good answer for you, but I'm thinking about it, and I hope that the community will insist when they submit a paper for publication that they refuse to take the methods out of the paper. And I've managed to do that a few times.
So there's a lot of sort of digested information. Say, for example, when you open the UCSC genome browser and you look at RNA-seq data, is that trustable or not?
I'm already in a lot of trouble, and I'm a little bit hesitant to answer that. I'll tell you what I do think. There's a feeling by especially biologists that it's very complicated to do RNA-seq analysis, that you need a lot of computers and a big analysis team, and that the processed result is so complicated that you better just trust it, because otherwise you can't do anything. And I don't believe that to be true. I feel that in my group, we can take we just took the Jovatis data set, and we can analyze a data set with a person or two. I think that, the right answer to your question is that, I'm personally skeptic of everything I read. When I read a methods paper, I ask myself, is it correct? And I think about what they're saying. In the same way, when you have a data set, I think you should download it, look at it, and maybe analyze part of it. You don't have to take all the samples, but run a program and quantify it. There's nothing particularly complicated about what any of these consortium are doing from my point of view. I'm sorry to say that, but that's what I feel. So I think the answer is yes, you should be skeptic, but not because what's at UCSC is bad, but just because the nature of the scientific method is not to trust blindly other people's work, but to think critically. So, yes.
Is there maybe one more? Okay, well, we're already 15 minutes into the poster session. So we got the poster session, we got cocktails, we got lots of time to keep discussing things. Thank you very much, Lior.
Thank you very much.